Hey everyone, welcome back to Blackfield, your trusted channel for exploring the wonders of science and nature. I am Astal Asbahan, and today we are diving into Unit 4 Genetics from our Grade 11 Biology curriculum. In this video, we will learn about genetic materials, the structure and function of DNA and RNA, cell division, and protein synthesis. Let's explore how our unique traits are passed from one generation to the next. Let's get started. Think about your family, your classmates, and everyone around you. Let's notice how we are all different. This difference comes from our genes, the blueprints of life. Genetics is the study of how traits are passed on through generations. Almost all organisms store their genetic information in DNA, although some viruses use RNA. This genetic material is the reason we look the way we do, and it controls how our bodies grow, develop, and function. DNA, or deoxyribonucleic acid, is the hereditary material in humans and many other organisms. Imagine a twisted ladder. That is what DNA looks like. The sides of the ladder are made of sugar, or deoxyribose, and phosphate groups, and the rungs are made of nitrogenous bases, adenine, thymine, guanine, and cytosine. These bases pair up like adenine with thymine and guanine with cytosine, forming the unique code that carries all the instructions for our body. Check out Figure 4.3 for visual explanation. Genes are segments of DNA that contain instructions for making proteins, and they determine our traits. These genes are organized into chromosomes. These chromosomes are tray-like structures made of DNA and proteins called histones. In diploid organisms like us, chromosomes come in pairs. For example, human cells contain 23 pairs, totaling 46 chromosomes. Figures 4.4 and 4.5 show how chromosomes look and how genes are arranged like birds on a string. DNA stores and transmits all the genetic information needed for life. Every time a cell divides, DNA must replicate itself in a process called DNA replication. This is a semi-conservative process, meaning each new DNA molecule has one original strand and one new strand. Enzymes such as DNA helicase, and DNA polymerase, DNA primase, DNA ligase, and topoisomerase work together to ensure that the genetic code is copied accurately. Figure 4.6 illustrates this process. RNA, or ribonucleic acid, is similar to DNA but has some key differences. RNA is single-stranded and uses the sugar ribose instead of deoxyribose. Instead of thymine, RNA has uracil. There are three major types of RNA. The first one is messenger RNA, which carries the genetic code from DNA to ribosomes. The second one is transfer RNA, which brings amino acid to the ribosomes during protein synthesis. The third one is ribosomal RNA, which combines with proteins to form ribosomes, which are the sites of protein assembly. Figure 4.7 shows the structure of RNA, the process of cell division. Cell division is essential for growth, repair, and reproduction. The process ensures that DNA is copied and distributed to daughter cells. There are two major types of cell divisions. The first one is mitosis, the second one is meiosis. Mitosis produces two identical daughter cells for growth and repair. Meiosis produces four genetically unique gametes for sexual reproduction. Figure 4.8 shows the cell cycle, highlighting how cells prepare for and undergo cell division. Here we have a detailed mitosis concepts. In mitosis, a single cell divides to form two identical daughter cells. This process includes prophase. Chromosomes condense and the nuclear envelope dissolves. During metaphase, chromosomes line up at the cell's center. Then during anaphase, sister chromatids are pulled apart to opposite poles. Then lastly, we have telophase and cytokinase step, which is new nuclear envelopes form and the cell divides. This ensures that each daughter cell receives an exact copy of the DNA. Here we have another detailed topic on meiosis. Meiosis is a special type of cell division that produces gametes, which refers to the sex cells. It happens in two stages. The first one is meiosis 1, the second one is meiosis 2. During meiosis 1, homologous chromosomes separate and crossing over occur, increasing genetic diversity. Then during meiosis 2, sister chromatids separate and similarly to mitosis. The result is four haploid cells, each with half the number of chromosomes, which contributes to the genetic variation. Figure 4.14 through 4.21 illustrates the stages of meiosis. Protein synthesis. Proteins are the building blocks of life, made up of amino acids. Protein synthesis is the process by which the genetic information in DNA is used to build proteins. This happens in two main steps. The first one is transcription, which is DNA is copied into messenger RNA in the nucleus. The second one is translation, which refers to messenger RNA travels to the ribosome in the cytoplasm, where transfer RNA brings amino acids to build a protein. The ribosome reads the messenger RNA code and assembles the amino acids in the correct order. Check out figure 4.22 and 4.24 to see how this process works. The central dogma of molecular biology. The flow of genetic information in a cell is known as central dogma, which refers to DNA change into RNA, then RNA to protein. This means that DNA is transcribed into RNA and RNA is translated into protein. Some viruses can even perform reverse transcription, converting RNA back into DNA. Figure 4.22 illustrates this fundamental concept. Here we have inquiry, activity, and reflection. Before we wrap up, here are some questions to think about. The first one is, what do you already know about DNA and RNA? Second one is, how does our genetic material make each person unique? The third one is, how do transcription and translation work together to produce proteins? In your group or individually, try to compare and contrast mitosis and meiosis by drawing their cycles and discussing the differences. Let me know your thoughts in the comments. To summarize today's lesson on genetics, genetics is the study of heredity and how traits are passed from parents to offspring. DNA, with its double helix structure, stores our genetic information, while RNA plays a key role in protein synthesis. Cell division through mitosis and meiosis ensures the genetic information is accurately transmitted and contributes to genetic diversity. Protein synthesis is protein synthesis via transcription and translation turns genetic instructions. That's it for today's deep dive into Unit 4 Genetics. I hope this video helped you understand how our genetic material works and how it controls everything from our traits to the production of proteins. If you enjoyed this video, please give it a thumbs up, share it with your friends, and subscribe to Blackpearl for more engaging science content. Feel free to drop your question or thoughts in the comments below. I would love to hear from you. Thanks for watching, and until next time, keep exploring and stay curious. Bye.